Something massive is floating in the dark, 1 mile underwater. No eyes, no tentacles. A body so thin you can see straight through it. And it just opened. Scientists call it the Deepstaria, a shape-shifting jellyfish with no face, no arms, and a secret passenger hiding inside its skin. In this video, we go into the deep. You'll meet a creature that turns itself inside out, a jellyfish that literally cannot die, and an octopus that picks its disguise based on who is hunting it. If you enjoy this, subscribe and stick around. Every chapter gets stranger than the last. Ready? We begin. The year is 1966. The Pacific Ocean, 300 miles off the California coast. Jacques Cousteau's research team is crammed inside a steel capsule the size of a minivan, dropping through total darkness. The vessel is called the Deep Star 4000, a brand new submersible built the previous year, designed to reach depths no diver could survive. The crew had done dozens of dives. They had seen strange things before: bioluminescent fish, pale eyeless shrimp, the usual parade of deep-sea oddities. Then the floodlights caught something that stopped everyone cold. Floating directly ahead was a massive translucent shape, pale, ghostly, pulsing slowly in the current like a lung breathing. The crew stared. Nobody spoke. The thing had no tentacles trailing below it, no visible face, no recognizable structure. It looked exactly like an enormous plastic bag drifting through open water, and it was easily wider than a man is tall. At first, the team assumed it was debris, a piece of equipment that had fallen from a surface ship. That assumption lasted about 30 seconds, because the thing moved. It contracted slightly, then expanded again, its edges curling inward and then rolling back out. The motion was deliberate, biological. Whatever this was, it was alive. Nobody on board could identify it. Cousteau's team had some of the most experienced ocean researchers in the world sitting in that capsule, and they were completely stumped. They filmed what they could, marked the location, and surfaced. The footage made it to a British marine scientist named Francis Russell the following year. Russell was one of the leading jellyfish experts alive at the time. He studied the footage, examined the limited physical description the crew had managed to record, and confirmed what the team suspected. This was a jellyfish, a species nobody had ever formally documented. He named the genus after the submarine that found it, Deepstaria, and the species itself he called Enigmatica, the Latin word for mystery. He was not being dramatic, he was being accurate. Here is what made it genuinely baffling. Every jellyfish known to science at that point used tentacles, long, trailing, venom-loaded strands that dangled below the bell and paralyzed anything that brushed against them. The tentacles were the whole strategy. They were the hunting tool, the defense system, and the feeding mechanism, all in one. Every single species, except this one. The Deepstaria had no tentacles at all, none. The entire bell was smooth, clean, exposed. Scientists had just discovered a jellyfish that had thrown away the one feature that defines a jellyfish as a hunter, and yet it was clearly thriving nearly a mile below the surface in one of the harshest environments on Earth. That detail alone should have triggered years of intensive research. Instead, the Deepstaria largely disappeared from scientific attention. The ocean is vast, the deep is expensive to explore. Follow-up dives at the same coordinates found nothing. The creature seemed to vanish as completely as it had appeared. For the next several decades, confirmed sightings could be counted on 2 hands. Scientists knew it existed. They had almost no idea how it worked. What Russell had no way of knowing in 1967 was that the mystery he had just named was far stranger than even the footage suggested, because the Deepstaria's most shocking feature had nothing to do with what it lacked. It had everything to do with what it could do. When scientists name a new species, the name usually tells you something. It describes a color, a shape, a location, or the person who found it. The name Deepstaria does all of that at once, and it still manages to undersell the strangeness of the animal. The genus name comes directly from the Deep Star 4000, the Cousteau submarine that made the original sighting. The species name, Enigmatica, is a direct label meaning mysterious or puzzling, so the official scientific classification of this creature translates roughly to the Deep Star Mystery. Scientists named it that in 1967. More than 50 years later, the name still fits perfectly. What makes that uncomfortable is that 50 years is a long time in biology. Scientists have sequenced the genomes of thousands of species. They have mapped the ocean floor with sonar systems precise enough to detect a hill 10 feet tall. They have built robotic submarines that can hover for hours and film in high definition at crushing depths. And yet the Deepstaria remains one of the least understood animals on Earth. Here is the core of the problem. The Deepstaria lives between 2,000 and nearly 6,000 feet below the surface. That zone, called the mesobathypelagic region, is dark, freezing cold, and under pressure intense enough to collapse a human lung instantly. Getting a submarine down there costs an enormous amount of money per hour of operation. Even when a vehicle reaches the right depth, finding a single Deepstaria in the open ocean is like trying to spot a single floating grocery bag somewhere in the Pacific from inside a car. In the entire 60 years since the first confirmed sighting, researchers have documented fewer than a few dozen verified encounters across the Pacific, Atlantic, and Indian Oceans. That number is staggeringly low for an animal that is over 3 feet wide when fully expanded. The Deepstaria is enormous by deep sea standards, and still it hides. The classification problem runs deeper than just rarity. Jellyfish belong to a group called Cyphozoans, and within that group they are sorted by their anatomy: bell shape, canal structure, reproductive organs. The Deepstaria fits the Cyphozoan category in some ways, and completely breaks the rules in others. Its lack of tentacles is the most obvious break. Every other member of its class uses them. The Deepstaria discarded that strategy entirely and replaced it with something that biology textbooks had no category for when the creature was first described. Scientists currently recognize 2 species, Deepstaria enigmatica, the ghostly white one found first, and Deepstaria reticulum, a deep red version formerly described in 1988 by a team of 3 researchers who found specimens in a separate ocean region. Same bagging behavior, same geometric internal structure, different color, slightly different pattern. 2 species in over half a century of searching. In an animal this large, that count suggests either an extremely low global population or a distribution so scattered that finding them requires pure luck. Every time a new submarine video surfaces, scientists treat it as a significant event. Each clip adds a small piece to a puzzle that should have been solved decades ago. The creature is real, documented, and classified, and somehow the ocean keeps it hidden with remarkable efficiency. The name might have been meant as a temporary label until more was understood. Instead, the mystery part turned out to be permanent. What no name captures is the physical reality of the animal itself, because to understand the Deepstaria, you have to understand what its body actually does. And that starts with the strangest anatomy in the ocean. Picture every jellyfish you have ever seen, at the beach, in a documentary, in a tank at an aquarium. The image is always the same: a dome-shaped bell floating upward, and below it a curtain of trailing tentacles, sometimes a foot long, sometimes 30 feet, depending on the species. Those tentacles are armed with millions of microscopic stingers. Touch one, and it fires a tiny harpoon loaded with venom. It is fast, it is automatic, and it works on almost anything that swims too close. The tentacle system is so effective that jellyfish have been using it for over 500 million years. They were hunting with it before dinosaurs existed, before trees existed, before anything with a backbone showed up on Earth. It is one of the most ancient and proven predator designs in the history of life. The Deepstaria has none of it. No tentacles of any length, no trailing strands, no stinging cells arranged in dangling ribbons. The bell sits alone in the water, smooth and unadorned, with nothing hanging below it. To a biologist seeing it for the first time, the reaction is something close to disbelief. An animal does not simply lose its primary survival tool over millions of years of evolution without replacing it with something equally powerful. So the immediate question becomes, what did the Deepstaria replace it with? The answer is the bell itself. In a standard jellyfish, the bell is basically a propulsion device. It pulses to push the animal through the water, and the tentacles trailing below do the actual work of catching food. The bell is transportation. The tentacles are the weapon. Deepstaria collapsed both functions into a single structure. The bell became the weapon, the trap, the stomach, all of it, wrapped into one shifting, expanding mass of translucent tissue. This is a dramatic departure from everything else in the jellyfish family tree. To pull it off, the Deepstaria evolved a bell with properties that no other jellyfish comes close to matching. The tissue is extraordinarily flexible. It can stretch to many times its resting size without tearing. It can contract just as rapidly, pulling inward from all sides simultaneously. The edges can curl and roll in ways that create a sealed enclosure. The bell also has no rigid parts, no hard sections, no skeletal support of any kind. It is entirely soft tissue from edge to center, which is part of why it can expand so dramatically and why it is so easy to destroy when handled. A robotic arm that grabs too firmly simply punctures it. Without tentacles, the Deepstaria also has no dedicated stinging defense against predators. It floats through the open ocean in total darkness, with nothing between its soft body and anything that wants to eat it. That vulnerability has puzzled researchers since the first description. How does a creature this exposed survive at depth for what is presumably a long lifespan? Part of the answer may be the darkness itself. At 2,000 feet down, very few predators are active in open water. The zone is too deep for most large fish and too cold for many aggressive hunters. The Deepstaria may have traded its tentacle defense for something far more valuable in that specific environment. A hunting strategy so efficient it barely needs to move. That strategy begins the moment the bell starts to open. And once you see how it works, you understand exactly why tentacles were never necessary. It starts as a small, dense shape. Imagine a crumpled paper bag, pale and translucent, drifting slowly through absolute darkness a mile below the surface. The Deepstaria at rest looks compact. Its bell folds in on itself, the edges overlapping, the tissue bunched together like fabric gathered into a fist. In that state, it could pass for a piece of floating debris. Then something swims nearby, the water shifts, and the transformation begins. The bell starts to open. Slowly at first, the edges peel apart and roll outward. Then faster, the tissue stretches in every direction at once, expanding radially like an umbrella opening in slow motion. What was a crumpled blob the size of a basketball becomes a wide, flat, billowing sheet 3 feet or more across. The expansion happens in seconds. The creature goes from compact to enormous faster than most fish can react. At full spread, the Deepstaria looks almost architectural. The bell becomes a broad, open canopy with thin, even walls stretched across a wide, circular frame. The tissue is so thin at this stage that light passes straight through it. The internal structure becomes visible from the outside. Veins of canals spread across the surface in a geometric pattern. The whole thing glows faintly against the dark water. The key to this transformation is the tissue itself. Most animal tissue has a limited stretch range. Muscle can extend to roughly 1.5 times its resting length before it tears. Skin has slightly more give. The Deepstaria's bell tissue operates at a completely different range. Scientists believe it can expand to many times its resting size in a single motion, without any structural damage. The material behaves more like a rubber membrane than like ordinary biological tissue. This is not purely a passive stretch either. The bell expands with directional control. The edges curl outward and downward simultaneously, which creates a cupped shape rather than a flat disc. That curl is critical. It means the expanded bell forms a bowl, a wide-open container, with the mouth facing downward and the edges curling slightly inward at the rim. Anything swimming beneath the deep staria at the moment of full expansion suddenly finds itself inside that bowl. The creature has not chased its prey. It has not stung anything. It simply opened around it. The water disturbance from the expansion itself may help drive small animals toward the center. Researchers have observed that as the bell opens, it generates a slow inward current at the edges, a gentle suction effect that pulls whatever is nearby toward the middle of the bowl. The prey effectively gets funneled in. Now here is where the shape becomes a trap. Once something is inside the open bell, the deep staria's next move is to close. The edges curl further inward. The bell contracts from all sides at once. The wide canopy collapses back toward the center, but now it seals at the opening rather than compressing into a dense blob. It closes like a bag being cinched shut. The prey is inside a sealed biological pouch with no exit. The deep staria has just performed a hunt using nothing but geometry and timing. What happens to the prey inside that sealed bell is where the story gets even stranger, because digesting a meal inside a body the size of a bedsheet requires a system unlike anything else in the animal kingdom. The bell has closed. The prey is inside. From the outside, the deep staria now looks like an inflated bag. The edges of the bell have pulled together and sealed, trapping water and the captured animal in a sealed internal chamber. No tentacles fired. No venom delivered. The prey is simply enclosed, swimming in circles inside a soft prison it cannot break through. The sealing mechanism itself confused researchers for years. Standard muscle tissue in most animals contracts along a single axis. A bicep shortens in one direction. A jaw muscle closes along one plane. The deep staria's bell edge closes in a coordinated ring, pulling inward from all points of the rim simultaneously. This requires the tissue to contract radially from every direction at once, like the opening of a camera lens running in reverse. Scientists studying footage frame by frame found that the seal happens in a single fluid motion. There is no hesitation, no partial closure that leaves a gap. The rim pulls shut completely and holds. For a creature with no brain and no central nervous system, that coordination is remarkable. The response is entirely driven by distributed nerve cells spread across the bell tissue, a simple web of signals that coordinates the whole structure without any central command. Once sealed, the prey cannot escape through muscle force alone. The bell tissue is flexible, but it resists puncture well. A small crustacean or fish inside the closed bell would need to tear through several inches of dense gelatinous material to break free. Most prey items are too small and too disoriented to manage that. They exhaust themselves trying. What happens next is dissolution. The deep staria does not swallow its prey in the way a fish swallows. The prey dissolves. The inner surface of the bell secretes digestive enzymes directly into the enclosed chamber, breaking down the captured animal while it is still inside the sealed space. The bell essentially becomes a stomach. The entire interior surface participates in digestion at once. This is a staggeringly efficient design. A standard animal digestive system moves food through a tube, processing it in stages. The deep staria surrounds its food from every angle simultaneously. The dissolved nutrients are then absorbed directly through the inner lining of the bell. But here is where an obvious problem arises. The bell, when fully expanded, covers a surface area roughly the size of a large pizza box. The tissue at the far edges of that surface is a long distance from the center where most digestion occurs. How do nutrients travel from the inner lining of the bell out to the extreme edges of the tissue? There is no heart pumping blood. There is no circulatory system in the mammal sense. The deep staria solved this with a structure that looks more like engineering than biology. Spread across the entire interior surface of the bell is a network of fluid-filled canals. These branching channels distribute the dissolved nutrients from the digestion zone out to every corner of the bell. The network fans out from a central point and splits repeatedly, reaching all the way to the outermost edges of the tissue. When lit from behind by submarine floodlights, those canals make the bell look like a stained glass window, a geometric web of lines crossing and branching across a glowing translucent surface. That network is the deep staria's answer to having a body too large to feed any other way, and it is far stranger up close than it sounds from a description. Hold a leaf up to sunlight. You can see the veins, fine lines branching from a central stem, splitting into smaller branches, reaching all the way to the outermost edge of the leaf. That branching pattern exists to move water and nutrients to every cell across a wide, thin surface. The deep staria's internal canal system works on the same principle at a much larger scale, inside living animal tissue. When a research submarine turns its floodlights directly onto a deep staria from behind, the effect is striking. The bell becomes backlit, and the canal network inside it becomes visible from the outside. The canals catch the light differently from the surrounding tissue. They glow faintly along their edges. The pattern that emerges looks almost mathematical, straight segments meeting at precise angles, branching outward from a central hub, dividing and dividing again, until thin terminal channels reach the far rim of the bell. Scientists have described it as resembling a cracked sheet of glass or a spider web stretched across a window. The canals are not symbolic, they are functional. After the deep staria dissolves a captured animal inside its sealed bell, the resulting nutrient-rich fluid needs to travel outward through the tissue to feed the entire structure. The bell can span more than 3 feet when open. Without a distribution system, the outermost sections of tissue would starve. No nutrient could reach them through passive diffusion across that distance in any reasonable time frame. The canal system solves this. Fluid moves through the channels, carrying dissolved nutrients outward from the digestion zone toward every point along the bell's surface. The flow is slow but continuous. Every region of the tissue receives a share of each meal. The deep staria, despite having a body that expands to the size of a dining table, wastes almost nothing. What makes this even more unusual is that the canals form during the animal's development and are permanently embedded in the bell tissue. They are structural. When the bell contracts to its resting size, the canals compress and fold along with the tissue. When the bell expands, the canals stretch back out, maintaining their geometry across the enlarged surface. The network is elastic in the same way the bell is elastic. No other known jellyfish has an internal canal system that performs this function at this scale. Other cyphozoans have simpler canal arrangements used for gas exchange or basic fluid movement. The deep staria's network appears to be a dedicated nutrient distribution system, purpose-built for a body plan that has no other way to feed itself across its full extent. Biologists also believe the canals may serve a secondary purpose. Because the bell tissue is so thin and the canals sit close to the outer surface, the canal fluid may help regulate the temperature of the tissue in the near-freezing water. Whether this is an incidental effect or an evolved function remains an open question. There is one more detail the canal network reveals when viewed on film. Deep inside the bell, at the center of the geometric web, researchers frequently spot something that should not be there. A small, dense object, usually bright crimson red, sitting motionless at the hub of the canal system. It is alive, it has legs, and it has been riding inside this creature for reasons nobody has fully explained. Science runs on samples. To properly understand an animal, researchers want tissue, they want genetic material, they want preserved specimens they can study under microscopes, slice into sections and analyze with chemistry. For almost every creature in the ocean, collecting a sample is a solved problem. A net, a jar, a suction tube, and the specimen is secured. With the deep staria, every standard method fails. The bell tissue is gelatinous and extraordinarily thin. When fully expanded, The walls of the bell in some sections are barely thicker than a few sheets of paper layered together. That thinness is what allows the extreme expansion, but it also means the tissue has almost no resistance to mechanical stress. Touch it with a rigid surface and it deforms. Apply any pressure beyond the lightest contact and it tears. Early attempts to collect Deepstaria specimens using standard net sampling produced the same result every time. By the time the net reached the surface, the jellyfish inside had disintegrated. The physical stress of the capture, combined with the pressure change as the submarine ascended from depth, shredded the tissue completely. Researchers arrived at the surface with a container of dissolved protein and a few fragments of canal material. Nothing usable. Robotic arms, which are the precision alternative to nets, created a different version of the same problem. The arms used on deep-sea research vehicles are designed to handle rock samples and equipment. Their grips are calibrated for solid objects. When operators attempted to collect a Deepstaria by grasping the bell edge with a robotic arm, the results were consistently destructive. The grip pressure, even at the lowest setting, punctured the tissue immediately. Several documented attempts on video ended with the bell tearing apart within seconds of contact. There is a famous piece of footage from 2012 captured when a research vehicle's thrusters came too close to a Deepstaria during a standard survey dive. The water jets from the thrusters meant for gentle repositioning of the submarine hit the Deepstaria directly. The force was enough to turn the entire creature inside out. The bell inverted completely. The canal network, previously visible only through the outer skin, suddenly faced outward. The creature pulsed slowly, apparently uninjured, and gradually righted itself over the following minutes. The video went viral because it looks almost violent. The creature collapses inward, flips, and reforms. But what it actually demonstrates is a terrifying fragility. A force gentle enough to barely disturb the surrounding water turned the animal inside out. Imagine what a robotic grip does. Because physical samples almost never survive collection, the genetic profile of the Deepstaria has only been partially assembled. Researchers have managed to extract small genetic sequences from fragments of tissue that survived collection long enough to be analyzed, but a complete genomic profile requires intact viable tissue. That tissue consistently fails to survive the journey to the surface. The fragility also means researchers cannot maintain a Deepstaria in captivity. No aquarium has ever held a living specimen. Every attempt to transport one has ended in dissolution. The animal exists only in its native environment, miles below the surface, in conditions that cost tens of thousands of dollars per hour to access. Every fact known about the Deepstaria has been learned by watching it on a screen through a camera mounted on a submarine. That limitation shapes everything science knows and does not know about this creature. And those pressure conditions it lives in are far more extreme than the fragility of its body suggests possible. Stand at the surface of the ocean. The weight of the entire atmosphere above you pushes down at about 14 pounds per square inch. You do not feel it because your body is built to handle exactly that amount. It is your normal. Now descend. Every 33 feet of water you drop adds another 14 pounds per square inch of pressure. By the time you reach 100 feet, the kind of depth recreational divers visit, the pressure has roughly tripled. Your lungs compress, your ears ache, your body starts working to equalize. The Deepstaria lives between 2,000 and nearly 6,000 feet below the surface. At 2,000 feet, the pressure pressing on every square inch of surface is roughly 60 times what you feel standing in open air. At the deeper end of its range near 6,000 feet, that number climbs to over 180 times surface pressure. That is enough force to crush a sealed metal container, enough to collapse an unprotected human chest in seconds. Steel submarines are built with pressure hulls inches thick to survive those depths. They are engineered with precise calculations accounting for every weld and joint. A failure in any section means implosion. The crew has no warning and no survival window. The Deepstaria floats through that same environment with tissue thinner than a sandwich bag, The reason it survives has to do with a fundamental principle of pressure physics. Pressure at depth crushes things by creating a force difference between the inside of an object and the outside. A sealed metal container collapses because the inside holds surface pressure air, while the outside is under crushing ocean pressure. The pressure difference tears it apart. A body with no sealed air spaces has no internal air pocket to be crushed. The deep staria is made almost entirely of water. Its tissue is mostly gelatinous fluid. Water does not compress meaningfully under pressure. At 6,000 feet, a water-based tissue does not experience a pressure difference between its inside and outside, because both sides are essentially the same incompressible fluid under the same load. The pressure does not crush it because there is nothing to create a difference between inside and outside. The creature is pressure neutral. It is in equilibrium with its environment at every depth it inhabits. This is the same principle that allows deep-sea fish to survive without being crushed. Their bodies contain no meaningful air spaces. Their swim bladders, if they have them, are filled with oil rather than gas. Everything is fluid. Everything equalizes. But the deep staria takes this further than almost any other creature its size. Because it is nearly all water and nearly no structure, it has almost zero rigidity. A rigid body at those depths creates stress points. Joints, bones, and shells all have junctions where pressure loads concentrate. The deep staria has no such points. It bends, deforms, and reforms around whatever force the water applies. This is why the submarine thruster that turned it inside out did not kill it. A body with no skeleton simply inverted. There was no frame to fracture, no joint to dislocate. It folded and then unfolded, the same way a wet paper bag does when you push it inward. The pressure it handles daily would kill every other creature in this video, and yet it does it with tissue so soft a human hand could tear it apart. What sits inside that tissue, though, is far less fragile. 2012. A research submarine is conducting a routine survey dive in deep water. The operators are repositioning the vehicle, adjusting its angle with short bursts from the thrusters. Standard procedure. The thrusters fire for less than 2 seconds. Nobody expects anything dramatic to happen. The camera is still rolling, and what it captures in the next 30 seconds becomes one of the most watched deep-sea clips in internet history. A deep staria is floating in the frame. The bell is partially open, slowly pulsing in that characteristic wobbly rhythm. The thruster burst from the submarine hits it directly. The jet of water, weak enough that it barely disturbs the surrounding ocean, reaches the deep staria's edge and keeps going. In one fluid motion, the entire bell inverts. The outer surface folds inward. The inner surface rolls outward. The canal network, normally visible only faintly through the transparent skin, suddenly faces the camera directly. For a few seconds, the creature is fully inside out. The geometry of the internal structure is exposed from the outside. The hub where the canals meet at the center is now facing forward. Then, over the course of about 2 minutes, the bell slowly corrects itself. It ripples. The edges shift. The inverted shape gradually rights itself back to normal, and the creature resumes its slow pulsing as if nothing happened. Researchers watching the footage were struck by 2 things simultaneously. First, the creature showed no apparent distress response. A thruster powerful enough to invert a body that size should have triggered some kind of flight reaction, some muscular contraction, or rapid movement away from the stimulus. The deep staria simply righted itself and continued. Second, the inversion demonstrated something important about the bell tissue that no prior footage had shown so clearly. The tissue is reversible, completely. The same flexibility that allows the bell to expand from a compact blob to a 3-foot canopy also allows it to fold in the opposite direction without any structural failure. The canal network maintained its shape during the inversion. The tissue stretched in reverse without tearing. When the creature righted itself, the canals snapped back into their correct orientation without any visible damage. This told biologists something fundamental about the architecture of the bell. There is no preferred direction built into the tissue structure. No side of the bell is reinforced differently from the other. The outer surface and inner surface have essentially the same mechanical properties. The creature can be turned inside out and back again with no lasting effect. This is a property that almost no other animal tissue shares. For context, try turning a basketball inside out. The material would tear immediately because rubber has a preferred geometry. It is built to face one direction. Deep Staria tissue is built to face either direction with equal ease. The viral spread of the video created an unexpected scientific benefit. Marine biologists who had never seen a live Deep Staria before contacted research institutions after watching it. Several researchers who had been working on related deep-sea species recognized structural details in the footage they had been puzzling over in their own specimens. The clip generated more academic attention to the Deep Staria genus than any formal paper published in the preceding 20 years. A single accidental thruster burst taught more about this animal than decades of careful planned observation. That says a great deal about how difficult it is to study something living this far from the surface. And yet, in almost every piece of footage ever captured of this creature, something else is visible. Something small. Something that has absolutely no business being inside a jellyfish. Every time a camera finds a Deep Staria, something else is visible. Deep inside the translucent bell, at the center of the canal network, sits a small, compact shape. It is roughly 3 inches long, it is bright red, sometimes deep crimson, and it has legs. Jointed, hooked legs, anchored into the inner tissue of the jellyfish. It is not drifting, it is holding on. This passenger is a deep-sea isopod, a crustacean related to the pillbugs you can find under rocks in any backyard. Its ocean relatives include species that grow to the size of footballs. The isopods found inside Deep Staria are smaller, but dense and heavily armored compared to the soft tissue they live inside. The visual contrast in the footage is jarring. The Deep Staria's bell is nearly colorless, white or very faintly tinted, transparent enough to see straight through. Against that pale background, the crimson isopod stands out like a warning light in a dark room. Every time researchers review new footage and spot that red shape through the bell wall, the reaction is the same. Immediate recognition. The hitchhiker is back. The isopod gets inside through the bell opening. When the Deep Staria opens its bell to hunt, the entrance gapes wide enough for a small crustacean to swim in. The isopod enters, positions itself at the interior hub, and hooks its legs into the tissue. When the bell closes around prey, the isopod is sealed inside, along with whatever the jellyfish has caught. What the isopod does next is where the debate begins. But setting aside the debate for a moment, the mechanics of how it stays attached are worth focusing on first. The isopod's legs end in curved hooks. These hooks catch on the canal tissue at the center of the bell and hold with enough grip to resist the movement of fluid and tissue during the Deep Staria's expansion and contraction cycles. When the bell opens and stretches to full size, the isopod stretches along with the tissue it is anchored to. When the bell closes and compresses, the isopod compresses back toward the center. It rides every shape change the jellyfish makes, never losing its grip. This is physically demanding for the isopod. The tissue it clings to is moving constantly, yet footage shows the isopod remaining stationary relative to the bell center through multiple expansion and contraction cycles. Its grip strength relative to its body weight must be considerable. Researchers first clearly identified the isopod as a separate species from the jellyfish in 2019, when the exploration vessel Nautilus captured footage detailed enough to distinguish the isopod's body segments and leg structure through the bell wall. Before that, the red shape had appeared in earlier footage, but was difficult to identify conclusively without high-definition resolution. The species of isopod found inside Deep Staria appears to be a generalist deep-sea crustacean, observed in other contexts at similar depths. But its consistent presence inside the Deep Staria Bell, across multiple sightings spanning different ocean basins, suggests the relationship is not accidental. This is not a random encounter. The isopod is choosing this home deliberately. Why it makes that choice, and what the arrangement costs or benefits the jellyfish, is one of the most genuinely unresolved questions in deep-sea biology. And the two leading theories about that question are more different from each other than almost any competing theories in recent marine science. Two scientists look at the same footage, same red isopod, same hooks dug into jellyfish tissue. They come to opposite conclusions. This is exactly where marine biology stands on the Deep Staria isopod relationship. The evidence supports both interpretations. Neither camp has the data to close the argument. The parasite case is built on anatomy. The isopod hooks its legs directly into the bell tissue. In footage, the attachment points show slight depression where the hooks anchor, meaning the legs are not just resting against the surface. They are embedded. Tissue that is being physically penetrated by another organism's limbs is, in the most basic biological sense, being damaged. Parasites routinely cause this kind of mechanical damage. A tick burrows its mouthparts into skin to feed. A lamprey uses hooked teeth to anchor while consuming blood. In both cases, the host tissue is violated for the parasite's benefit. Researchers arguing the parasite interpretation point to the hook embedment as evidence the isopod is actively feeding on the jellyfish tissue itself, slowly consuming it from the inside, while staying hidden from any predator that would otherwise target a free-swimming crustacean in open water. This theory has a certain elegance. The isopod gains shelter in one of the most protected environments at that depth. No predator will reach into a living Deep Staria bell to extract a hidden crustacean. The jellyfish, meanwhile, is gradually depleted. The symbiosis case rests on a different reading of the same facts. The isopod is consistently positioned at the center of the bell, near the hub where the canal network originates. When the jellyfish dissolves prey inside the sealed bell, fragments of that dissolved material travel through the fluid environment toward the canal openings. The isopod, sitting at that hub, is perfectly positioned to intercept small fragments of the digested prey. Under this interpretation, the isopod is scavenging. It is eating the scraps of whatever the jellyfish catches, without harming the jellyfish itself. In exchange, some researchers speculate the isopod may provide a benefit. It might clean the inner surface of the bell, consuming bacteria or small organisms that would otherwise settle on the tissue. It might help break down larger prey pieces that the jellyfish enzymes struggle to dissolve quickly. This theory requires the jellyfish to be tolerating the isopod's presence, which is possible if the isopod's benefits outweigh the cost of feeding it. Many deep-sea organisms maintain exactly this kind of low-cost exchange. The problem is that neither theory can be properly tested with current technology. Confirming parasitism requires demonstrating that the isopod consumes jellyfish tissue at a rate that harms the host. Confirming symbiosis requires demonstrating that the jellyfish receives a measurable benefit. Both require long-term observation of the same individual pair over time. A Deep Staria in its natural environment is only ever observed during the duration of a single submarine dive, typically a few hours at most. Scientists cannot tag the jellyfish and return to it later. The deep ocean makes that kind of follow-up effectively impossible. So the debate continues with each new footage release. Researchers study the isopod's position. They estimate body mass and food intake. They calculate whether the jellyfish shows signs of tissue degradation at the attachment points. And they publish papers that reach different conclusions. The answer, when it finally comes, will probably require technology that does not yet exist. Until then, the isopod rides. What it eats while riding, however, is somewhat clearer. The isopod is sealed inside a living creature. The bell has closed. Whatever prey the Deep Staria captured is dissolving around it, and the isopod is exactly where it wants to be. To understand what the isopod eats, you first have to understand the environment inside the sealed bell. during active digestion. The deep staria secretes digestive enzymes through the inner lining of the bell. These enzymes break down the captured prey into smaller and smaller pieces. The process is gradual, not instantaneous. A small crustacean or fish inside the closed bell might take hours to fully dissolve. During that time, particles and fragments of the prey are suspended in the fluid filling the interior space. The isopod, positioned at the hub of the canal network at the center of the bell, sits directly at the point where this nutrient-rich fluid is most concentrated before it gets distributed outward through the canals. It has front-row access to every meal the jellyfish catches. Its mouthparts are designed for this situation. Deep-sea isopods are primarily scavengers and omnivores. Their feeding appendages can process a wide range of food types, from solid fragments to dissolved organic material. Inside the deep staria bell, the isopod likely feeds on both. Larger fragments of prey that have not fully dissolved get consumed directly. The fluid surrounding those fragments provides dissolved organic material the isopod can absorb. This feeding strategy is remarkably efficient. The isopod expends almost no energy hunting. It stays anchored, waits for the jellyfish to catch something, and then feeds from the resulting material without ever exposing itself to the open water where it would need to compete, evade predators, and spend enormous amounts of energy just moving. The energetic math of this arrangement favors the isopod completely. At depths where food is scarce and every calorie matters, a creature that can access a reliable food source without hunting has an enormous survival advantage. The deep staria essentially acts as a food delivery system. But the arrangement also creates a timing dependency. The isopod can only eat when the jellyfish catches something. If the jellyfish goes through a long period without a successful hunt, the isopod goes without food as well. Researchers believe the isopod can tolerate extended fasting periods, as most deep-sea crustaceans have evolved to go without food for long stretches given the unpredictability of the deep ocean food supply. There is also the question of oxygen. Inside a sealed bell, the oxygen dissolved in the trapped water will be consumed by the prey's decomposition and the isopod's own respiration. Scientists estimate that for short hunting events, the oxygen supply inside the bell is sufficient. The jellyfish likely releases the seal and reopens its bell periodically, even between active hunts, which would refresh the interior water supply and prevent oxygen depletion. One additional detail observed in footage is the isopod's behavior when the bell is open. The isopod does not leave. Even when the bell opens fully and the path to open water is unobstructed, the isopod remains anchored at the center. This consistency suggests the isopod has no reason to leave, or that the risk of open water is high enough that staying inside an apex predator's bell is worth any downsides. That level of commitment raises an obvious follow-up. Is this particular isopod a permanent resident? Does it live its entire life inside one jellyfish? Or does it move between hosts? Science has no confirmed answer. Every single deep staria encounter lasts only as long as a submarine can maintain position, and the creature drifts away before any long-term behavioral pattern can be established. What is clear is that two separate species of deep staria have both been found hosting these passengers, the ghostly white one and the deep red one, which raises the question of how two jellyfish this similar ended up looking so completely different. For the first 22 years after Francis Russell named the deep staria enigmatica, science believed there was only one species, one ghostly, white tentacle-free shape-shifting jellyfish in the deep ocean. One mystery. Then in 1988, a team of 3 marine biologists published a formal description of a second species from the same genus. They named it Deep Staria reticulum. The two species share the same basic design. Both have the shape-shifting bell. Both lack tentacles. Both use the bagging strategy to capture prey. Both have the geometric canal network spread across the bell interior. Both have been found hosting the red isopod passenger. But stand the two species side by side in footage, And the difference is immediate. The Enigmatica is pale, almost colorless, translucent white, with a faint milky quality that makes it look like a ghost floating in the dark water. Its canal network, when backlit, glows clearly against that pale background. The reticulum is deep red, in some footage, almost burgundy. The color saturates the entire bell, from the rim to the center. The canal pattern on the reticulum is slightly different as well, with a coarser geometric structure and wider channel spacing compared to the finer, denser network of the Enigmatica. Why the two species developed such dramatically different coloration is genuinely unknown. Color in deep-sea animals usually serves a specific function. Red is actually a useful camouflage color at depth, because red light wavelengths do not penetrate to those depths. An animal that is red in shallow water looks bright and visible. An animal that is red at 2,000 feet down appears nearly black to other creatures' eyes, because there is no red light available to reflect off its surface. Red effectively becomes invisible at depth. The reticulum may have evolved its deep red color as a camouflage adaptation. If other animals in its environment perceive it as dark or invisible, it gains a hunting advantage. An expanded bell that appears as a dark, featureless shadow in the water would be less likely to trigger an avoidance response in potential prey. The Enigmatica's pale white coloration is harder to explain using the same logic. White reflects whatever light is available, which at 2,000 feet is mostly bioluminescent light from other organisms. A pale bell that reflects bioluminescence might actually glow faintly, making it more visible rather than less. Some researchers speculate that the Enigmatica's transparency serves as its camouflage. An animal with almost no visual opacity is harder to detect than one with a solid color, even a dark one. A transparent bell expanded in open water might be close to invisible to the limited visual systems of deep-sea prey animals. Both species inhabit the same depth range and appear across the same general ocean regions. Whether they compete for the same prey in the same zones, or whether they occupy slightly different microhabitats within that depth range, remains unconfirmed. Sighting records are too sparse to map territory. What is confirmed is that 60 years of searching has turned up only 2 species in the entire Deepstaria genus. 2. For an animal this large, in 3 separate ocean basins, that number suggests either a global population that is genuinely very small, or a distribution strategy that keeps individuals so spread apart that encounters are rare even for submarines actively searching for them. That rarity has a cascading effect on everything scientists want to know about the creature's life cycle, because there is one stage of that life cycle that has never, in over half a century of research, been observed at all. Every animal reproduces. That is not a small statement. Reproduction is the mechanism through which every trait, every adaptation, and every survival strategy gets passed to the next generation. Understanding how an animal reproduces tells you its population structure, its growth rate, its vulnerability to environmental change, and how it evolved in the first place. For the Deepstaria, scientists know none of this. No researcher has ever recorded a Deepstaria mating. No one has ever identified a juvenile Deepstaria. No larval form has been collected and confirmed to belong to this species. The complete life cycle from the moment of fertilization to the development of a mature adult is entirely undocumented. This is an extraordinary gap for a species discovered in 1966. Over 5 decades of oceanic research, with increasingly sophisticated equipment, and the fundamental biological cycle of this animal remains invisible to science. Part of the explanation is logistical. The Deepstaria lives in a zone that costs enormous resources to access. A research vehicle operating at 2,000 feet burns through time and money at a rate that makes extended observation prohibitively expensive. A submarine crew that finds a Deepstaria during a dive might have a few hours to observe it before the dive window closes. Following a single animal through any part of a reproductive sequence would require days of sustained observation in the same location. The fragility problem compounds this. Even if researchers could observe a mating event or locate juveniles, collecting the specimens for analysis would likely destroy them. The tissue that makes adult Deepstaria impossible to transport intact would make juveniles even more fragile. A larval jellyfish is typically microscopic and gel-like. Bringing one to the surface without degradation would require containment technology specifically designed for that purpose, and no such system has been deployed for Deepstaria. Most cyphozoan jellyfish follow a well-documented life cycle pattern. Adult males and females release eggs and sperm into the water. The fertilized eggs develop into a free-swimming larval form. Those larvae eventually attach to a hard surface on the sea floor, transforming into a stationary polyp. The polyp grows and eventually releases juvenile jellyfish called ephori, which mature into adults. The full cycle takes months to years depending on the species. Whether the Deepstaria follows this same cycle or has evolved an alternative strategy is unknown. The depth at which it lives creates complications for the standard cycle. The seafloor at those depths is cold, dark, and under crushing pressure. Whether a Deepstaria larva could successfully attach, develop into a polyp, and release ephori in those conditions is a question with no data behind it. Some researchers have suggested the Deepstaria might reproduce through a modified version of the standard cycle, with the polyp stage. Others have proposed that the Deepstaria might skip the polyp stage entirely and reproduce through direct development, a strategy seen in some other deep-sea invertebrates. No evidence exists for either proposal. The population implications of this ignorance are significant. Without knowing the reproductive rate, scientists cannot assess whether the Deepstaria population is stable, growing, or declining. They cannot identify what environmental pressures might threaten it. They cannot determine how climate-driven changes in ocean temperature or chemistry might affect its breeding cycle. A species that large, in 3 ocean basins, with a life cycle invisible to science, represents a fundamental blind spot, and the implications of that blind spot extend far beyond understanding one jellyfish. What happens to a Deepstaria after its life ends, however, has recently become one of the more surprising discoveries associated with this animal. The Deepstaria is not immortal. Whatever its lifespan, it ends. The bell stops pulsing, the tissue goes still, and then, slowly, the body begins to sink. For decades, scientists assumed jellyfish were poor contributors to the deep-sea food web after death. A jellyfish body is mostly water. Its tissue is thin and gelatinous. The assumption was that it would decompose quickly in the water column as it sank. Dissolving long before it reached the seafloor, the nutrients would disperse into the open water, contributing nothing to the organisms living on the bottom. That assumption turned out to be wrong. Research submarine dives over the past 2 decades have captured footage of what scientists now call jelly falls. A jellyfish body, sometimes the body of a Deepstaria, reaches the seafloor and lands with its full structure largely intact. The cold temperatures of the deep water slow decomposition dramatically. The low bacterial activity at extreme depth slows it further. The body arrives at the bottom in a state that makes it a concentrated food source. What follows is immediate and intense. Within minutes of a jelly fall landing on the seafloor, organisms begin to arrive. Crabs emerge from the sediment. Shrimp converge from the surrounding darkness. Amphipods, small crustacean scavengers, arrive in swarms. These animals rarely have access to food sources this large in a single location. The deep seafloor at those depths is a near desert, with organic material arriving mostly as a slow drizzle of tiny particles from above. A whole Deepstaria body landing in that environment is the equivalent of a feast appearing in a place that normally offers scattered crumbs. The footage of jelly falls shows animals feeding in a coordinated frenzy that is unusual for deep-sea organisms. Normally, deep-sea scavengers are spread thin across the seafloor, foraging independently for scarce food. Around a jelly fall, dozens of individuals cluster and feed simultaneously. The geometric tissue of the Deepstaria bell that canal-laced structure that was built for nutrient distribution in life, gets consumed systematically from the edges inward. Researchers have estimated that a single large deep staria carcass can sustain a cluster of deep-sea scavengers for several days before the tissue is fully consumed. Given how little food is available in those zones, several days of concentrated feeding represents a meaningful caloric event for every animal involved. This discovery changed how marine biologists view jellyfish in ocean ecosystem models. If jellyfish bodies routinely reach the seafloor intact and deliver concentrated nutrition to bottom-dwelling communities, then jellyfish blooms at the surface have a direct downstream effect on deep seafloor ecosystems. More jellyfish at the surface means more jelly falls. More jelly falls means better fed benthic communities at extreme depth. For the deep staria specifically, the jellyfall research suggests the animal plays a larger ecological role than its rarity would imply, even with only a few dozen confirmed sightings over 60 years. If each individual deep staria contributes a jellyfall event upon its death, the cumulative effect across 3 ocean basins over decades adds up to a significant nutrient contribution. The isopod inside the bell faces an interesting question when its host dies. Whether it abandons the sinking body before it reaches the seafloor, or rides it down and becomes part of the jellyfall scavenging event, has not been captured on film. The answer might say a great deal about whether the isopod's relationship to the host extends beyond the animal's living state. There is one more chapter in the deep staria story that belongs to the scavengers, though, because the feeding event that follows a jellyfall is dramatic enough to qualify as its own discovery. The body is on the seafloor. The canal network, stretched and geometric in life, has collapsed into a flat mass of tissue against the sediment. The isopod, if it stayed, is buried beneath the weight of the bell. The surrounding water is freezing, close to 30 degrees Fahrenheit, and nearly without motion. Then the first crab arrives. Deep-sea crabs in this zone are slow movers by the standards of shallow water species. Cold temperatures reduce metabolic rates, which reduces movement speed. A deep-sea crab on the move covers ground at a pace a slow walk could outpace. But chemical signals travel through water efficiently, and the dissolved proteins from a deep staria carcass spread outward through the still deep water like a signal fire. Within minutes of the body landing, the first scavengers reach it. Within an hour, footage shows dozens of organisms clustered around the site. The visual is striking. Around the pale, flat remains of the bell, crabs crowd in from multiple directions, their legs overlapping as they jostle for position. Shrimp swarm in the water directly above, waiting for fragments that get dislodged and drift upward. Amphipods, smaller than a fingernail, cover the tissue surface in dense clusters, working through the gel with specialized mouthparts designed for exactly this kind of soft organic material. The canal network that made the deep staria's bell distinctive in life creates a particular pattern in death. The canals are slightly denser than the surrounding tissue, made of a slightly firmer gel. Scavengers consume the thinner sections of the bell first, working between the canal lines. The canal structure itself takes longer to break down and remains visible for hours after the surrounding tissue has been cleared. For a time, the jellyfall site looks like an excavation, with the geometric canal line standing slightly above the sediment, while everything around them has been stripped clean. The full consumption of a large deep staria carcass takes multiple days. By the end, the site contains almost nothing recognizable. The tissue is gone. Even the toughest structural parts of the bell have been processed. The sediment at the site is enriched with the waste products of the feeding event, adding nutrients to the seafloor that will slowly filter into the sediment and support microbial communities for weeks afterward. Each feeding event at a jellyfall also supports population dynamics among the scavengers. Crabs and other deep-sea invertebrates require periodic high-calorie food events to support reproduction. The steady trickle of marine snow, the fine particles of organic material that drift down from the surface, maintains basic survival. But a jellyfall provides enough concentrated nutrition to fuel reproductive cycles. Populations that regularly benefit from jellyfalls show higher reproductive success than those in areas where jellyfalls are rare. The deep staria's ecological role, therefore, runs in two directions. During its life, it functions as a predator in the deep pelagic zone, removing small crustaceans and fish from the water column. After its death, it functions as a nutrient delivery event for the seafloor community below. It feeds things both as a hunter and as a carcass. For an animal so rarely seen, that dual contribution suggests a significance that the raw sighting count does not reflect. Each confirmed deep staria in the ocean represents an active ecological node, alive or dead, contributing to a food web that operates in a zone humans have only barely begun to map. What brought public attention to that hidden food web, though, was something far less scientific. It was a video that millions of people watched and completely misidentified. In 2012, a video clip appeared online and spread rapidly across news sites, social platforms, and science blogs. The title on most posts was some variation of "Mysterious Sea Creature Caught on Camera by Oil Rig." The footage showed a massive, translucent shape in deep water, pulsing slowly, its edges rippling in an almost deliberate rhythm. The internal structure, the geometric canal network, glowed faintly under the rig's floodlights. The comment sections filled within hours. People identified the shape as an alien organism, a mutant jellyfish created by deep-sea oil contamination, a creature from a science fiction film that had somehow appeared in real ocean footage, a government-controlled deep-sea drone, a ghost. Several posts claimed the video proved unknown megafauna lived in the ocean's deepest zones, organisms large enough to rival whales, but never catalogued by science. None of those explanations were correct. The footage was a deep staria. Specifically, it captured a deep staria in the partially open phase of its expansion cycle, filmed by the remotely operated cameras mounted on an oil rig's subsea infrastructure. The rig's lights provided backlighting that made the canal network unusually visible, which contributed to the alien quality of the footage. The pulsing motion that so many viewers found disturbing was the ordinary contraction rhythm of the bell tissue. Marine biologists identified the creature within days of the video's spread. The identification was not difficult for anyone familiar with deep staria footage. The canal network geometry, the tentacle-free bell, and the characteristic expansion rhythm are all distinctive. A few researchers published brief online explanations. Most of the internet ignored those corrections and continued sharing the alien sea monster version. The scientific community's reaction to the viral event was mixed. Some researchers found the widespread misidentification frustrating, a symptom of how little public understanding exists of deep-sea biology. Others recognized an opportunity. The video's reach brought genuine public curiosity about deep-sea organisms to a scale that years of academic publishing had never achieved. Several marine biology programs used the footage in their courses as a case study in how rapidly misinformation spreads when legitimate scientific context is absent. The deep staria's actual biology, once explained, proved compelling enough to hold student attention on its own merits. A real creature that turns itself inside out and hosts a crimson hitchhiker is genuinely more interesting than a fabricated alien. The oil rig context also raised a legitimate scientific point. The footage demonstrated that deep staria individuals sometimes drift into proximity with subsea industrial infrastructure. Oil rigs and offshore platforms operate in deep water zones that overlap with deep staria habitat. The remotely operated vehicles used in routine rig maintenance create video records of the surrounding water. Those records represent an unintentional monitoring system for deep-sea fauna. Several researchers began reviewing archived footage from oil rig operations after the viral event, looking for additional deep staria sightings and other deep-sea animal encounters that had been captured incidentally during maintenance dives. The review produced additional confirmed deep staria sightings that had gone unrecognized at the time of filming. Operators who had flagged the footage as containing an unidentified object and moved on now had context for what they had seen. The internet's fascination with a misidentified jellyfish inadvertently expanded the confirmed sighting record for one of the rarest deep-sea animals known to science. A creature that hid from researchers for decades was finally getting found in archival footage because millions of curious people watched a confusing video and asked what it was. The answer, when it came, was stranger than any of the theories the comment sections had proposed. Before the 2012 viral moment, there was a longer and quieter history of misidentification, one that stretched back decades and cost science years of potential research time. The first people to encounter deep staria on a regular basis were not scientists, they were commercial fishermen and deep-sea trawling crews operating in the Pacific and Atlantic. These crews dragged enormous nets through the water column at significant depths, targeting fish populations in the mesopelagic zone. Occasionally, something unusual came up in the catch. When a deep staria came up in a trawl net, it came up destroyed. The mechanical stress of the net, combined with the pressure change during ascent, shredded the bell tissue completely. What arrived on the sorting deck was not a recognizable animal. It was a mass of pale, thin, translucent material tangled in the net mesh, gelatinous, featureless. Every crew that encountered this material reached the same conclusion: plastic. Specifically, a piece of a discarded fishing net or a large plastic bag that had drifted to depth and been swept up during the trawl. The material was thrown back without a second thought. No record was made. No sample was saved. This happened across multiple fishing fleets, in multiple ocean regions, over multiple decades. The deep staria was landing on fishing boats and being discarded as litter. The species had been encountered dozens of times before its formal discovery, and every single time it was misidentified as garbage and returned to the sea. The irony is significant. One of the rarest animals in the deep ocean, with a body plan unlike anything else in marine biology, was being dismissed as human waste. And the very feature that makes the creature so biologically remarkable, its extreme translucency, and the featureless quality of its contracted bell, is what made it look so convincingly like a piece of floating plastic. Even after the 1966 discovery and the 1967 formal description, the problem persisted. Trawling crews were not routinely informed about the existence of a translucent, tentacle-free jellyfish they might encounter. The academic description of the deep staria existed in scientific journals that fishing crews did not read. The disconnect between discovery and practical field recognition lasted for years. Some researchers believe this misidentification pattern means the deep staria's historical range and population were significantly underestimated. Every discarded specimen that was thrown back as plastic represents a data point that was never recorded. The sighting record that makes the deep staria appear extremely rare might be partially an artifact of this identification failure. There may have been far more encounters than the official record reflects. The plastic misidentification also points to a broader challenge in deep-sea biology. Animals adapted to the deepest and darkest zones often have transparent, colorless, or featureless body forms. Transparency is a camouflage strategy at depth, but that same transparency makes these animals look to an untrained eye like inert debris. Valuable data slips away because the observer's reference framework does not include the possibility that the object is alive. Training deep-sea observers, including commercial fishing crews, to recognize and report unusual biological material became a priority for some marine research organizations following the deep staria's growing profile in the 2010s. The argument was straightforward. More eyes in the deep ocean produce more sightings. More sightings produce better population data. Better population data eventually answers some of the questions that controlled research dives have failed to answer in 60 years. The deep staria was hiding in plain sight for decades on the decks of fishing boats, and nobody knew it. What was being discovered in the open ocean during those same decades, though, was a completely different kind of shapeshifter, one that used behavior rather than anatomy to become something else entirely. 1998, the shallow coastal waters off Sulawesi, an island in Indonesia. A marine biology team is filming on a sandy seafloor at about 60 feet of depth. Standard survey work. Tropical reef environments are well studied, and the team expects familiar species. Then an octopus emerges from the sand and does something no octopus has ever been documented doing. It flattens its 8 arms against its body, stiffens them into a rigid bundle, and begins undulating them in a side-to-side wave. Its skin shifts to bold brown and white stripes. Its body elongates. The shape it creates matches a banded sea snake almost perfectly. The posture, the movement, the coloration, all of it together produces an imitation detailed enough to make the filming team look twice. They had just recorded the first documented sighting of the mimic octopus. Every octopus can change color. The skin of all octopus species contains specialized cells called chromatophores, tiny sacs of pigment that expand and contract under muscular control, changing the visible color and pattern of the skin in less than a second. Octopuses use this ability to blend into coral, rock, and sand. It is one of the most sophisticated camouflage systems in the animal kingdom. The mimic octopus does all of that, and then goes several steps further. Changing color is passive camouflage. The mimic octopus changes its posture, its movement pattern, its three-dimensional shape, and its behavior simultaneously to actively impersonate another species. The distinction matters enormously. Blending into a background is about disappearing. Impersonating a toxic animal is about becoming a threat. The mimic octopus's full repertoire, documented across multiple research observations, includes accurate impersonations of over 15 distinct species. The banded sea snake is one of the most common. It also impersonates the flatfish, spreading and flattening its body while swimming just above the seafloor in the characteristic rippling motion of a flounder. It impersonates the lionfish by spreading its arms radially and holding them stiff and spine-like, mimicking the venomous spines that make lionfish nearly untouchable. Each of these animals is genuinely dangerous to predators. Banded sea snakes carry venom potent enough to kill a human. Lionfish spines inject a toxin that causes severe pain and tissue damage. Flatfish are poor targets for most predators due to their flat, hard-to-swallow shape. The mimic octopus has built a library of impersonations specifically targeting animals that predators already know to avoid. What makes this genuinely extraordinary is the selection process. Research observations have documented that the mimic octopus does not randomly rotate through its repertoire. It appears to choose which animal to impersonate based on the specific predator threatening it. When confronted by a damselfish, which is territorial and aggressive toward intruders in its reef patch, the mimic octopus preferentially impersonates the banded sea snake, which is a known predator of damselfish. The mimic deploys the one disguise most likely to frighten the specific animal in front of it. This level of behavioral flexibility suggests a cognitive process more complex than simple reflex. The octopus is assessing the threat, selecting from multiple available responses, and executing a physically demanding imitation. All of it happens in seconds. The deep staria shapeshifts its body to survive a physical challenge. The mimic octopus shapeshifts its identity to survive a social one. Both strategies work in their respective environments with remarkable success. But one animal in the ocean takes shapeshifting further than either of them, and it does it at the level of the cell itself. The damselfish is aggressive and territorial. It guards a small patch of reef with fierce consistency, attacking anything that enters its zone. Other fish know this and give damselfish territories a wide berth. The mimic octopus does not avoid those territories, it moves through them. And when the damselfish charges, the octopus responds in a way that is almost calculated. In documented footage, the mimic octopus facing a damselfish does not display all 15 of its known impersonations. It displays the banded sea snake. Specifically, the banded sea snake is one of the few animals in the reef environment that actively preys on damselfish. The mimic octopus appears to know this. How an octopus could develop this specificity is a question that marine biologists have worked on since the species was formally described in 2001, 3 years after the first sighting. The answer likely involves a combination of learning and hardwired response. Young mimic octopuses may learn which disguises work against which threats through repeated encounters during development. Over time, those pairings become fast, reflexive responses. The execution of each disguise requires precise physical control. To impersonate a flatfish, the mimic octopus compresses its body, spreads its mantle flat, holds all 8 arms in a specific arrangement, and moves across the seafloor in a rippling glide that matches the swimming motion of a flounder. Each component of that impersonation requires independent motor control. The octopus is coordinating 8 limbs, its mantle shape, its skin color and pattern, and its movement speed simultaneously. Octopuses have a distributed nervous system. Over half their neurons are located in their arms rather than in their central brain. Each arm has a degree of autonomous processing ability. This architecture may be what allows the mimic octopus to simultaneously control so many independent physical variables during an impersonation. The arms can handle their own positioning, while the central brain manages color, pattern, and movement direction. The lionfish impersonation is particularly demanding. The mimic octopus spreads all 8 arms radially away from its body and holds them stiff, pointing outward and slightly upward. Real lionfish spines are rigid and fixed. The octopus's arms are flexible. Holding flexible appendages rigid in specific positions requires sustained muscular effort, the equivalent of holding a difficult yoga pose while also managing skin color and remaining aware of an approaching threat. Researchers studying the mimic octopus have also noted that it is not infinitely flexible. There appear to be physical limits to its repertoire. The animals it impersonates are all relatively flat or elongated species. The octopus cannot impersonate a bulky, round-bodied animal because its own body plan cannot produce that shape. Its disguises are constrained by its own anatomy, which means the 15+ species in its repertoire are all animals it can physically approximate. This constraint actually supports the cognitive selection argument. If the octopus was simply running through random impersonations, it would occasionally attempt shapes it cannot physically produce, and produce incoherent results. The fact that its entire documented repertoire consists of animals it can credibly imitate suggests it does not attempt shapes it cannot achieve. The mimic octopus shapeshifts through behavior. The Deepstaria shapeshifts through tissue mechanics. Both are extraordinary solutions to the challenge of survival in an environment where nothing stays still and everything is a potential threat. The next creature on this list shapeshifts in a way that breaks the biological rules both of those animals follow, because it does it at the level of individual cells, and it uses that ability to escape the one thing every other animal cannot escape. Every living thing ages, cells divide, accumulate damage, and eventually stop working. Tissues break down, organs fail, the machinery of life runs its course and stops. This is not a flaw in biology. It is a fundamental feature of how multicellular life operates. Cells divide a limited number of times, programmed decline is built into the system, and death follows. One jellyfish decided not to follow that program. Turritopsis dohrnii is tiny. Fully grown, it reaches about 1/5 of an inch across, smaller than a pinky fingernail. Its bell is transparent, its tentacles thin and numerous, and its most visible feature is a bright red stomach visible through the clear bell wall. It lives in warm, temperate ocean waters across the world. If you passed one while swimming, you would not notice it, but when this jellyfish is injured, starving, or has simply reached old age, something happens that has no parallel anywhere else in the animal kingdom. The adult jellyfish sinks to the sea floor, its bell collapses inward, the tentacles retract, the pulsing stops. To any observer, the animal appears to be dying. and then the cells begin to change. The process is called transdifferentiation. Mature, fully specialized cells, cells that have already become muscle tissue or nerve tissue or skin cells, revert to an unspecialized state. They lose their specific function and become something closer to what they were at the earliest stage of the animal's development. From that unspecialized state, they then redevelop into a completely different cell type. A muscle cell becomes a nerve cell. A skin cell becomes a digestive cell. The entire cellular identity of the organism shuffles. The result is that the adult jellyfish physically transforms back into a juvenile polyp. The organism anchors itself to a surface, looks and functions like a newborn version of itself, and then restarts its entire life cycle from the beginning. It grows back into a medusa, the adult jellyfish form, and lives again. This cycle has no confirmed upper limit. Researchers who have studied Turritopsis dornii in laboratory settings have observed the reversion cycle occur multiple times in the same individual. The jellyfish aged, reverted, regrew, aged again, and reverted again. The process appeared stable. There was no degradation in the reverted form across cycles. Each reversion produced a juvenile that grew into a fully functional adult. The theoretical implication is that this jellyfish cannot die from aging. External threats, being eaten, physical damage, disease, can still kill it. But the cellular clock that drives aging in every other multicellular organism does not appear to run the same way in Turritopsis dornii. It can reset that clock whenever conditions require it. The mechanism behind transdifferentiation in this species has been partially mapped by genetic researchers. The jellyfish appears to regulate a set of genes that control cell identity in a way that most animals cannot. In typical animals, once a cell commits to a specific identity during development, that commitment is locked in. The Deepstaria cannot reverse that lock. The mimic octopus cannot reverse that lock. Turritopsis dornii can. The discovery of this ability was initially met with skepticism. Cellular reversion at this scale was thought to be biologically impossible outside of cancer, where cells lose their identity in a destructive rather than controlled way. The Turritopsis dornii reversion is orderly, directed, productive. It is the opposite of cancer. Where cancer represents cells losing control of their identity, this jellyfish demonstrates cells changing identity on purpose. How it controls that process at the molecular level is still being investigated, and the answer might eventually matter far beyond jellyfish biology. The reversal does not happen randomly. Something triggers it. Researchers have identified multiple triggers that initiate the transdifferentiation process in Turritopsis dornii. Physical damage to the bell, prolonged starvation beyond the animal's tolerance threshold, environmental stress such as sudden temperature changes or salinity shifts, and simply reaching the end of the natural adult lifespan. All of these conditions send the animal into the reversion sequence. The trigger appears to activate a specific set of genes. These genes, which are normally suppressed in the adult form, begin to express themselves when the stress signal arrives. Their expression initiates the deconstruction of the adult body form and the cellular reprogramming process. What follows, at the cellular level, is genuinely difficult to describe without sounding like science fiction. Muscle cells stop contracting and begin changing their gene expression profile. The proteins that make them muscle cells are gradually replaced by proteins characteristic of a different cell type. The cell physically restructures. Its internal architecture changes. Its relationship to neighboring cells changes. The muscle cell has become something else. This happens across the entire organism simultaneously, thousands of cells undergoing identity changes in a coordinated fashion. The bell dissolves its structure. The tentacles are reabsorbed. The adult body is deconstructed from the inside, while the cellular material is preserved and redirected. The end product is a cyst-like structure that attaches to a surface. Inside that structure, the reprogrammed cells begin organizing into the tissue layers of a juvenile polyp. The polyp develops. Buds form. new juvenile jellyfish are released. The cycle begins again. Researchers confirmed this sequence through laboratory observation, where individual jellyfish were tracked through complete reversion cycles under controlled conditions. They also confirmed it genetically by showing that the adult and reverted forms of the same individual carry identical genetic material. The reverted polyp is not an offspring, it is the original animal restructured. The implications for broader biology have generated significant research interest. Transdifferentiation, the conversion of one specialized cell type to another, is a process that scientists in regenerative medicine have been trying to achieve in mammalian cells for decades. The ability to convert one type of adult cell into another without going through an embryonic state would have profound applications for tissue repair and disease treatment. In most animals, transdifferentiation either does not occur naturally or occurs in very limited forms. Some salamanders can regrow lost limbs through a process that involves partial dedifferentiation of cells at the wound site, but that is a localized event affecting a small number of cells. The Turritopsis dohrnii reversion involves every cell in the organism simultaneously. The genes that control this process in the jellyfish have evolutionary relatives in mammals, including humans. The same gene families that manage cell identity and development in jellyfish are present in human cells. What differs is the regulatory machinery that controls when and how those genes express themselves. In humans and other mammals, the system is locked in a way that prevents adult cells from reverting to earlier states. Whether studying the jellyfish's regulatory mechanism could provide a roadmap for unlocking similar processes in mammalian cells is a genuinely open scientific question. Several research groups have published work on the genetic similarities between Turritopsis dohrnii's reversion genes and human developmental genes. The field is active and ongoing. The immortal jellyfish resets itself with a molecular switch. A limpet discovered just months ago does something physically similar, but from the outside rather than the inside, reshaping its own skeleton to match wherever it lives. October 2025, a research vessel is conducting a survey dive off the coast of Japan, exploring a seafloor zone near hydrocarbon seeps at about 3,000 feet below the surface. Hydrocarbon seeps are areas where methane and other chemicals leak up from beneath the seafloor, supporting unusual communities of organisms that live on chemical energy rather than sunlight. These environments consistently produce biological surprises. The research team's cameras are scanning the base of a bacterial mat when they spot something on a cluster of worm tubes. A small shell, pale and translucent with a faint blue-white tint, clinging to the exterior of the tube structure. It is a limpet. Limpets are common mollusks found in tide pools and shallow waters worldwide. A cone-shaped shell, a muscular foot gripping the surface, a slow-moving grazer of algae and bacteria. Familiar animals. Except this shell is wrong. The typical limpet shell is a simple cone, symmetrical, slightly curved. The same basic shape whether the limpet is sitting on a flat rock, a curved mussel shell, or a vertical cliff face. The shell grows in one shape and stays that way regardless of where the animal lives. This shell is cylindrical, tall, narrow, and elongated in a way that mirrors the worm tube it is sitting on. The team looks at neighboring limpets on different surfaces. One clinging to a flat mussel shell has a flattened, disc-like shell. One on the side of a rock outcrop has a shape somewhere between the two. Same species, completely different shells. The team collected specimens and brought them to the surface for analysis. The new species was formerly named Pyropelta artemis, but researchers also began calling it the Artemis limpet after the precision and adaptability the name suggests. The finding was published in early 2026, drawing immediate attention from both the mollusk biology community and the broader field of biomechanics. What the Artemis limpet does is reshape its shell to match the geometry of whatever surface it attaches to during its juvenile development phase. The shell grows in a direction and shape that mirrors the contact surface. On a cylindrical worm tube, the shell grows upward and narrow, matching the tube's vertical geometry. On a flat mussel shell, the shell grows outward and flat, matching the horizontal surface. The limpet's shell architecture responds to environmental geometry and adjusts accordingly. This is a form of phenotypic plasticity, the ability of an organism to change its physical form in response to environmental conditions without changing its underlying genes. Many animals display limited versions of this. Plants grow toward light sources. Some fish develop different jaw shapes depending on the prey available in their habitat. But skeletal restructuring at this level, where the hard, calcified shell itself grows into a fundamentally different three-dimensional shape based on environmental geometry, is exceptionally rare in any organism. The practical benefit is clear. A shell shaped to its attachment surface creates a more secure fit. A cylindrical shell wrapped around a cylindrical tube has more contact area with the surface it grips than a standard cone shape would. More contact area means a stronger hold against currents and predator attacks. The limpet is essentially custom-fitting its armor to its home. What makes this discovery land differently than a typical new species announcement is the timing. Within months of each other, researchers described a limpet that reshapes its skeleton and confirmed details about a jellyfish that cannot die of old age. Both animals reshape themselves in response to circumstances. Both do it with a precision that took scientists decades to even notice, let alone explain. And the Artemis limpet raises a question that applies to all the shapeshifters in this video. The shell of most mollusks grows in one direction. Calcium carbonate is deposited at the shell's growing edge, layer by layer, expanding the shell outward from a central point in a predictable spiral or cone. The geometry is fixed by genetics. A scallop always grows a scallop shape. A cone snail always grows a cone. The form is determined before the animal ever encounters its environment. The Artemis limpet breaks that pattern at the point of initial attachment. When the juvenile limpet first settles on a surface, its shell has not yet developed beyond a microscopic starting structure. The contact surface it attaches to begins influencing the direction of shell growth almost immediately. Researchers analyzing specimens from different surface types found that the divergence in shell shape begins within the first few weeks of the animal's life and continues consistently through its full development. The mechanism appears to involve mechanoreceptors in the limpet's foot, sensory cells that detect the shape and texture of the surface the animal is pressing against. Those signals are processed and translated into changes in the direction of calcium deposition at the shell's growing edge. When the surface curves upward, when the surface is flat, the shell grows flat. The limpet's body reads the geometry of its home and instructs its shell accordingly. This sensing and response system requires a feedback loop between the limpet's nervous system and its shell-building tissue that researchers are still mapping. The shell-building cells, called the mantle, produce calcium carbonate in controlled layers. For those layers to grow in a geometry-matching direction, the mantle tissue must receive directional information from the foot's mechanoreceptors and use that information to bias where and how fast calcium is deposited along different sections of the shell edge. The precision of the result is remarkable. Specimens collected from worm tube environments have shell shapes that fit their tubes closely enough to look intentionally manufactured. The cylindrical form matches the tube's outer diameter accurately. The shell wall thickness is distributed evenly around the circumference. If you did not know these were biological structures, you might assume they had been machined to fit. The benefit extends beyond grip strength. A shell that fits its surface precisely also reduces the gap between the shell edge and the attachment surface. That gap is a vulnerability. Predators that prey on limpets typically insert something, a limb, a proboscis, a specialized mouthpart, into the gap between the shell and the surface to pry or probe the animal out. A minimal gap is a genuine defensive advantage. The Artemis limpet's custom-fit shell reduces that gap to near zero along its entire contact edge. Researchers have proposed that this adaptation is specific to the hydrocarbon seepy environment where the species was found. Seep environments are structurally complex, with worm tube fields, mussel beds, bacterial mats, and carbonate rock formations all occurring in close proximity. An animal that can attach successfully to any of these surface types without requiring each to match a preset shell geometry has a significant habitat flexibility advantage over a species locked into one shell shape. That flexibility is precisely what connects the Artemis limpet to the Deepstaria and the mimic octopus. All three animals thrive in environments that demand physical adaptation. All three produce that adaptation through mechanisms that most animal bodies are not built to perform. The question that naturally follows is whether this kind of flexibility is becoming more common in the ocean, or whether these animals are ancient survivors of an older biological strategy that modern science is only now discovering. The Deepstaria has existed for an unknown period of time. Its fossil record is essentially non-existent because gelatinous soft-bodied animals almost never preserve as fossils. The Turritopsis dornii, the immortal jellyfish, belongs to a group of animals called hydrozoans that have a fossil record stretching back hundreds of millions of years. The mimic octopus's lineage is younger, but still ancient. These are not new strategies. Shape-shifting in various forms has been a biological tool for a very long time. But marine biologists studying these animals have noted a pattern that is harder to explain away as coincidence. The rate at which new shape-shifting species are being discovered and described has accelerated in recent decades. The Artemis limpet was found in 2025. The mimic octopus was formally described in 2001. The immortal jellyfish's reversion ability was confirmed in the 1990s. Before that, the known catalogue of significant biological shapeshifters was thin. Part of this acceleration is technological. Deep-sea cameras and remotely operated vehicles have only reached their current resolution and depth capabilities in the past few decades. Many animals that were known to exist could not be studied behaviorally until high-definition underwater video became standard equipment on research vessels. The mimic octopus had probably been living in Indonesian waters long before 1998. Nobody had filmed it clearly enough to document its behavior. But some researchers argue that environmental pressure may also be accelerating the behavioral expression of shape-shifting abilities that were previously less intensively used. Ocean temperatures are rising, prey distributions are shifting, habitat boundaries are moving. Animals with rigid body plans or fixed behavioral responses face increasing mismatch between their capabilities and their environment. Animals with high phenotypic plasticity, the ability to adjust their physical form or behavior in response to environmental change, maintain function across a broader range of conditions. Under this interpretation, the shapeshifters are not just ancient survivors. They may be thriving because the ocean is changing in ways that reward flexibility. A Deepstaria that can expand to whatever size the prey situation demands is less constrained by prey size shifts than a tentacled predator locked into a fixed hunting strike range. An Artemis limpet that can grip any surface type is less vulnerable to habitat structural changes than a species that only fits one geometry. The counter-argument is that these animals are not new. They evolved their abilities in stable ancient oceans, and their current success owes more to those deep evolutionary roots than to any recent environmental advantage. The shape-shifting was always there. Humans just recently developed the tools to notice it. Both arguments have scientific support, and no definitive test exists to distinguish between them with current data. What is agreed upon is that phenotypic flexibility represents a category of biological capability that has been consistently underestimated in the historical classification of marine species. The ocean is far larger and far more varied in depth, temperature, and chemistry than any terrestrial environment. Organisms that can adjust to that variety without needing to evolve new structures over millions of years have an enormous adaptive advantage. The creatures in this video, the Deepstaria, the mimic octopus, the immortal jellyfish, the Artemis limpet, represent extreme cases of that principle. Each one pushed the flexibility of its body plan further than any close relative. That extreme flexibility is most impressive when you understand what the environment that produced it actually looks like. 2,000 feet straight down, the sunlight stopped penetrating the water about 1,000 feet above you. For the past 1,000 feet, the descent has been through total darkness. The water temperature has dropped steadily since you passed the thermocline, the boundary layer where warm surface water meets cold deep water. Down here, it hovers just above freezing, about 34 degrees Fahrenheit. The pressure is 60 times what you feel at the surface. Every square inch of your body would receive 60 pounds of force from every direction simultaneously. The air spaces in a human body, lungs, sinuses, middle ear, would be compressed and crushed. Without a rigid pressure suit or a steel submersible hull, survival at this depth is measured in fractions of a second. The water is still, not calm in the way a lake is calm on a quiet morning, still in a more absolute sense. There are no waves here, no wind, no tidal turbulence. The only water movement comes from deep ocean currents that shift slowly over days and weeks rather than seconds. A piece of floating material at this depth moves so slowly it appears stationary. There is no food drifting by in obvious form. The food that reaches this zone arrives as marine snow, tiny particles of organic material, dead plankton, bacteria, fecal pellets, shed skin cells that drift downward from the sunlit surface zone above. By the time marine snow reaches 2,000 feet, much of its nutritional content has been consumed by organisms in the intermediate zones. What arrives is sparse and dilute. An animal that filters water for food at this depth gets very little from each liter it processes. The darkness is not the soft darkness of a moonless night. It is the complete absence of light. Human eyes, given years to adapt, would detect nothing. The only light present is bioluminescence, the cold glow produced by organisms that generate their own light through chemical reactions. Bioluminescence serves multiple functions at depth. Some animals use it to attract prey, some use it to signal mates, some use it as a defense, flashing light to startle predators, or illuminate attackers for their own predators to see. The deep staria produces no bioluminescence. In the dark, it is effectively invisible. A 3-foot-wide animal with no light output in a lightless environment is genuinely undetectable at any range greater than the reach of a submarine's floodlights. Navigation at these depths happens chemically. Most deep-sea predators and scavengers locate food through dissolved chemical signals in the water. They detect amino acids and lipids shed by prey, follow the concentration gradient upward, and find the source. This is slower than visual hunting, but works across much larger distances in an environment where visibility is zero. The animals that live in this zone have adapted in ways that make them look, to human eyes, like creatures from a completely different biology. Many are transparent, many produce their own light, many have enormous eyes relative to their body size, maximized for detecting any available photons. Many have expandable stomachs, capable of swallowing prey larger than their own bodies, because food encounters are so rare that missing an opportunity to feed could mean weeks without nutrition. The deep staria's strategy makes complete sense in this context. An animal that can expand to 3 feet and trap anything that wanders inside does not need to chase prey or compete for a hunting position. It waits. In an environment with almost no energy to spare, that patience is not laziness. It is precision engineering. And yet, this environment, this hostile and extraordinary place, is where the deep staria hunts without any of the tools that every other deep-sea predator has developed to survive. Every major deep-sea predator has a version of the same toolkit: teeth, or a beak, or a rigid structure designed to seize prey physically, eyes or pressure sensors, or electrical field detectors to locate prey in darkness, speed or stealth, or a lure designed to bring prey within reach. The anglerfish dangles a bioluminescent lure. The viperfish has needle-like teeth designed to trap fish that swim into its path. The giant squid uses 2 elongated tentacles that shoot outward faster than most cameras can track. The Deepstaria has none of this. No teeth, no rigid structures, no bioluminescent lure, no speed, no tentacles, no specialized sensory organs that researchers have been able to identify beyond the basic distributed nerve net present in all jellyfish. It drifts, it opens, it waits. This passive hunting approach is called ambush predation, but the Deepstaria takes it to an unusual extreme even within that category. Most ambush predators maintain a fixed position and strike when prey enters their range. A trapjaw spider waits on its web. An anglerfish holds station and lures prey toward it. The strike, when it comes, is fast. The prey has to be prevented from escaping before it can react. The Deepstaria's version of ambush predation removes the fast strike entirely. The bell expands slowly by predator standards. The expansion takes several seconds. Most fish capable of swimming at depth could detect the water movement from a bell, expanding 3 feet wide, and simply swim away before the trap closes. The organisms the Deepstaria primarily targets likely cannot do this. Researchers believe its prey consists mainly of small crustaceans, copepods, and other zooplankton that drift passively or have limited directional swimming ability. These animals cannot detect and respond to the slow expansion of a Deepstaria bell fast enough to escape before the edges curl inward. The water displacement from the expansion may also work against the prey in a counterintuitive way. As the bell opens and the volume inside increases, water flows inward toward the center from all directions to fill the expanding space. Small animals caught in that inward flow get pulled toward the center of the bell, exactly opposite to the escape direction. The Deepstaria's hunt might be partially self-reinforcing. The act of opening generates the current that draws prey inward. This would explain why the creature can be an effective predator at depths where active swimmers are rare and the energy cost of chasing prey would be prohibitive. If the bell's expansion itself assists in concentrating prey inside, the Deepstaria barely needs to do anything other than open. The energy economics of this strategy are favorable in a way that becomes clearer when you consider the alternative. A deep-sea predator that actively chases prey burns calories on the chase. If the chase fails, those calories are lost. The Deepstaria expends minimal energy expanding its bell, waits, and either captures something or contracts back to its resting state and drifts until the next opportunity. Failed hunts cost almost nothing. For an animal living where food is scarce and every calorie matters, a hunting strategy that loses almost no energy on failure is about as efficient as biology gets. There is one further mystery about this hunter. Despite its efficiency, its numbers are extraordinarily low. The global population of Deepstaria across 3 ocean basins remains genuinely unknown, but the sighting record over 60 years suggests it is smaller than a creature this effective at surviving should produce. That population mystery may be the most unsettling fact about the Deepstaria. 60 years, 3 oceans, dozens of research programs with access to deep-sea vehicles, and the confirmed sighting record for the Deepstaria sits at fewer documented encounters than most people have birthdays in their lifetime. This number demands an explanation. An animal over 3 feet wide, floating in open water with no camouflage beyond transparency, should not be this difficult to find repeatedly. Transparency works in total darkness, but research submarines carry powerful floodlights that illuminate the surrounding water in a sphere of visibility wide enough to spot a Deepstaria several yards away. Every dive in the right depth range is essentially a search with a moving spotlight, and still the encounters are rare enough to be treated as events worth publishing. Several explanations have been proposed, and none of them fully resolves the paradox. The first is distribution. The mesobathypelagic zone, the depth range the Deepstaria inhabits, covers an enormous volume of water. The Pacific Ocean alone contains enough water in that depth band to fill billions of Olympic swimming pools. Research submarines explore a tiny fraction of that volume on any given dive. Even an animal present in relatively healthy numbers would be statistically unlikely to appear in the narrow cone of illuminated water a submarine sweeps during a standard dive. The second explanation involves the deep staria's drift behavior. Unlike active swimmers that cover predictable distances based on their speed and direction, the deep staria moves entirely with the currents. Deep ocean currents, at its depth range, are slow, complex, and three-dimensional. Deep staria individuals could be concentrated in specific current gyres or distributed thinly across vast open stretches. Without current mapping combined with population surveys, finding deep staria becomes partly a question of being in the right current system at the right time. The third explanation is the one researchers find most concerning. The global deep staria population may genuinely be small. The combination of unknown reproductive rate, fragile tissue that makes adults vulnerable to physical damage, and a life cycle that is completely undocumented creates the possibility that this species maintains a low population equilibrium. If the deep staria reproduces slowly, or if juvenile mortality is high due to the extreme conditions of its habitat, the adult population at any given time might be measured in the thousands across 3 ocean basins, rather than the millions. For a species with no commercial value and no representation in any fisheries management system, a small and potentially declining population could go unnoticed for decades. There are no monitoring programs targeting the deep staria, no population surveys, no tagging studies. The absence of sightings is not tracked as a data point anywhere. The deep staria could be declining and science would not detect that trend from the current sighting record, because the baseline was never established precisely enough to measure change against. The misidentification history makes this worse. If deep staria were routinely caught in trawl nets and discarded as plastic for decades, those encounters represent a mortality source that was never counted. Each destroyed specimen was a potential adult removed from the population with no record. What the population situation actually is remains unknown. It might be stable and simply distributed in ways that make encounters rare. It might be in decline and nobody has the data to confirm it. The ocean is large enough to hide both possibilities indefinitely. That ambiguity itself reveals something significant about how much of the deep ocean is still genuinely unexplored. The deep staria is large, 3 feet wide when expanded. That is the width of a standard doorway. An animal that size, floating in the ocean, was completely unknown to formal science until 1966, and it was found by accident. A submarine happened to be in the right place. The crew happened to film it. The footage happened to reach someone who could identify it as Mew. Remove any of those coincidences, and the deep staria might have remained unknown for additional decades. This raises an uncomfortable question about everything else that might be down there. Marine biologists estimate that the deep ocean represents by far the largest habitable zone on Earth by volume. The sunlit surface layer where most ocean life is studied makes up a small fraction of the total ocean volume. The mesopelagic and bathypelagic zones, the depth ranges below where sunlight penetrates, contain the majority of the ocean's water volume. They also represent the zone where human exploration is most limited by cost, technology, and physical challenge. Current estimates from the scientific community suggest that the deep ocean floor alone has been directly observed and mapped with high-resolution imagery over less than a small fraction of its total area. The open water column below 1,000 feet has been surveyed even less systematically. The number of species living in those zones that have never been encountered by humans is genuinely unknown, because you cannot count what you have never seen. What the deep staria demonstrates is that size is no protection against anonymity in the deep ocean. If an animal the size of a doorway can go undiscovered until the 1960s and remain poorly understood, 60 years later, the range of things that might exist in deeper, less explored zones is essentially unconstrained by conventional expectations. Deep-sea biologists have encountered other large unknown organisms on surveys. Footage from deep remotely operated vehicle dives routinely captures animals that researchers cannot immediately identify. Some of these turn out to be known species seen from unusual angles or in unusual states. Some represent genuinely new records. Occasionally, something appears that requires extended analysis to classify. The history of deep-sea exploration is filled with animals that were thought impossible or purely mythological before being formally described. Giant squid were considered sea monster legends, until carcasses began washing ashore and were eventually photographed alive in the deep ocean in 2004. The colossal squid, larger still, was formally described in 1925 but not photographed alive until much later. Both animals were large enough that their existence should have been obvious, and both remained in the realm of rumor for far longer than their size would suggest. The Deepstaria adds to this list an animal that was so strange in its design that when it appeared on the deck of a fishing boat, fishermen could not identify it as an animal at all. Its body plan was so far outside the expected template that it looked like debris. How many other organisms in the deep ocean have body plans equally foreign to human intuition? How many have been encountered and dismissed, or have never been encountered at all because they occupy zones that research vehicles visit infrequently, or never? The answer to those questions depends heavily on the technology used to explore the deep ocean, and that technology, despite decades of improvement, still fails in specific and important ways. The cameras mounted on modern deep-sea research vehicles are extraordinary pieces of engineering. High-definition sensors capable of capturing fine detail in zero ambient light, powered by their own light sources, mounted on robotic platforms that can hover at precise depths for hours. The footage they produce looks like science fiction compared to the grainy, low-resolution images from early submarine expeditions. And they are still not enough to answer the most basic questions about the Deepstaria. The core problem is encounter rate. The most advanced camera system in the world produces no data about a species it never sees. Research vehicles operate on dive schedules determined by funding, weather, and research priorities. A dive targeting a specific location might last 8 to 12 hours. The probability of encountering a Deepstaria during any given dive, even in regions where previous sightings have occurred, is low. Most dives in the right depth range produce no Deepstaria sightings at all. Autonomous underwater vehicles, which can operate for much longer periods without a crew, represent an improvement in search time. These robotic platforms can be programmed to follow survey patterns across large areas and record continuously. Several research programs have deployed autonomous vehicles in depth ranges where Deepstaria have been sighted. The longer coverage time has increased encounter probability, but the vehicles face a different limitation. Their programmed survey paths do not adjust based on real-time biological observations, the way a crewed vehicle can. If an autonomous vehicle passes near a Deepstaria and its cameras catch a brief image, the vehicle continues its programmed path, rather than pausing to observe. The sample collection problem remains completely unsolved. Every method attempted to bring a Deepstaria specimen to the surface intact has failed. Scientists have designed specialized soft suction collection devices, essentially large syringes with flexible tips, intended to draw a small tissue sample from the bell without contacting it directly. The pressure differential required to create suction deforms the tissue before the sample can be secured. The tissue folds, the suction tip contacts the bell surface, and the specimen is damaged. Some researchers have proposed developing a containment system that preserves the pressure conditions of the deep ocean during ascent, preventing the sample from experiencing the pressure gradient that causes disintegration. Such a system would require maintaining the pressure equivalent of 2,000 feet of water inside a chamber throughout a 10-hour ascent. The engineering challenge is significant, and no operational system has been deployed for this purpose. Genetic sequencing, which has transformed the study of inaccessible or rare species in other contexts, requires viable tissue. Environmental DNA sampling, a technique where genetic material shed into the water by living organisms is collected from water samples, has been applied to some deep-sea species with some success. For the Deepstaria, water samples from depth ranges where sightings have occurred have yielded fragmentary genetic sequences that confirm the animal's presence in those zones. But environmental genetic material degrades quickly at the temperatures and pressures of the deep ocean, and the sequences recovered are too incomplete for full genomic analysis. The practical result is that the Deepstaria remains studied primarily through video footage. Every major discovery about its behavior, its canal structure, the isopod relationship, the inside-out footage, came from someone watching a screen and observing what a camera captured during a limited dive window. That footage-only approach to studying an animal is extraordinary in modern biology. We know the gross anatomy of the Deepstaria from the outside. We have almost no data from the inside, and the inside, it turns out, is where the most important evolutionary questions about this animal live. An animal body, in the conventional framework of biology, is a solution to a specific set of problems: how to move, how to eat, how to avoid being eaten, how to find a mate and reproduce. Every structure in the body, every organ, every limb, every layer of tissue exists because it solved one or more of those problems well enough to persist across generations. The Deepstaria challenges that framework at almost every point. Its body has no rigid structures to provide movement. It does not chase prey. Its defense against predators is debated. Its reproduction is undocumented, and yet it persists in one of the harshest environments on Earth, presumably for millions of years, given how deeply embedded it is in the jellyfish family tree. The Deepstaria's body is the solution to a different version of those problems than biologists typically work with. Movement in a near still water column is achieved not by swimming, but by drifting. A body with no rigid parts has no drag resistance to fight. Eating is achieved by expanding to surround food, rather than pursuing it. Defense relies on being invisible in darkness rather than being fast or armored or toxic. These solutions look from the outside like a body that abandoned all of its functions, but they represent a complete, coherent alternative architecture for surviving in that specific environment. The mimic octopus makes the same point from a behavioral direction. Its body is a conventional cephalopod body, tentacles and a mantle and a well-developed nervous system. But its behavioral architecture, the ability to reprogram its appearance and movement on demand, creates an animal that functionally transcends its body plan. The octopus uses a standard body to achieve outcomes that no standard body plan should be able to produce. The immortal jellyfish takes the challenge further still. Animal bodies age because cellular machinery degrades over time and cannot be replaced indefinitely. This is treated as a universal constraint. Turritopsis dornii identified a pathway around that constraint by converting the degraded machinery back to its original state. The body becomes the tool that rebuilds itself. The boundary between life stages, which biology treats as a one-way transition, becomes reversible. The Artemis limpet questions whether a skeleton needs to be a fixed structure at all. Skeletons provide structural support. They do not, in the conventional understanding, need to match their environment's geometry. The limpet discovered that a skeleton which does match the environment's geometry provides better support and better defense than a standard one. The body form itself became a sensor and responder. Each of these animals represents a case where the rules of animal body design were pushed into a domain where they produced something that standard biology classes do not prepare students to expect. This has practical consequences for how scientists search for life in extreme environments, including environments beyond Earth. The assumption that life requires certain recognizable structural features, rigid bodies, defined organ systems, centralized nervous systems, is an assumption built from the sample of life visible at Earth's surface. The deep ocean demonstrates that life can persist and thrive with none of those features in their standard forms. Every time the deep ocean produces an animal like the deep staria, it expands the definition of what a living body can be, and that expanded definition matters for every search, scientific or otherwise, for life in environments that seem too extreme to support it. Which brings the question of the deep ocean's unexplored regions into sharper focus than ever. The ocean floor covers more than 70% of Earth's surface. The total area of the deep seafloor is larger than all the land on Earth combined. Scientists have directly imaged, with resolution high enough to identify geological and biological features, a fraction of that total area. The rest has been mapped with sonar, which shows elevation but not biology, or has never been studied at all. The areas that have been visited directly are not evenly distributed. Research programs concentrate around zones of scientific interest, hydrothermal vents, which support unique ecosystems, the margins of continental shelves, which are accessible at shallower depths, and regions near established research stations where logistics are manageable. The open abyssal plain, the vast flat stretches of seafloor between geological features, receives far less attention despite covering most of the seafloor's total area. The abyssal plain sits below the deep staria's depth range, generally below 6,000 feet, and extends down to the deepest trenches at over 35,000 feet in places like the Mariana Trench. Organisms adapted to those depths experience conditions beyond what even the deep staria handles. Pressures at the bottom of the Mariana Trench are over 1,000 times surface pressure. Temperatures are near freezing. The food supply is even sparser than at the deep staria's depth. And life is there, consistent with the biological reality that if a habitat exists long enough, something evolves to fill it. Researchers who have sent cameras and collection devices to the deepest trenches have found crustaceans, fish, worms, microbes in the sediment, sea cucumbers in quantities that suggest active populations. These are known organisms adapted to extreme conditions. What remains unknown is whether those depths also host animals with body plans as foreign to standard biology as the deep staria is to standard jellyfish biology. The history of deep-sea exploration argues strongly that they do. The pattern is consistent. Every time humans reach a new depth zone with adequate imaging technology, species appear that do not fit existing categories cleanly. The deep staria fit no existing jellyfish category in 1966. It still strains classification today. The immortal jellyfish performed a biological process that was considered impossible before it was documented. The Artemis limpet grew a different skeleton than any mollusk biologist expected to find. These surprises do not appear to be running out. The 2025 Artemis limpet discovery came from a depth range that research vehicles visit regularly. It was not found in an extreme deep zone. It was discovered because a camera happened to be in the right location, with adequate resolution to notice that a limpet's shell shape was unusual. How many animals in less visited depth zones are waiting for that same combination of presence and attention? The number is genuinely unconstrained. There is no basis for assuming the surprises stop at any particular depth or in any particular zone. The ocean is simply too large and too poorly explored for that assumption to be anything more than wishful comfort. The deep staria was the answer to a question nobody thought to ask until 1966. What is floating in the open water column at 2,000 feet, with no tentacles and a body that can expand to the size of a doorway? The answer turned out to be a creature with an isopod passenger, a geometric internal network, and a hunting strategy built entirely out of geometry. That answer raised more questions than it resolved. It always does. A single submarine, a team of scientists, one patch of dark water off the California coast, and a pale pulsing shape that nobody could explain. That was 1966. The deep staria was the answer the ocean gave when humans first started looking properly at depth. One species, tentacle-free, carrying a red crustacean in its belly and digesting prey with a geometric network carved into its own tissue. 60 years of research later, the most fundamental questions about this animal remain open. How it reproduces, how many there are, how long they live, whether the isopod is a partner or a parasite, whether the two known species compete or avoid each other. The list of unknowns for a single documented species is long enough to fill decades of future research programs. And the Deepstaria is the known one. What that means, placed next to the Artemis limpet found in 2025, the immortal jellyfish whose cellular reset was confirmed in the 1990s, and the mimic octopus documented in 1998, is a pattern. The ocean has been producing biological surprises at a consistent rate across every decade of serious deep-sea exploration. Each discovery arrives from a zone that was considered mapped, or from a species considered understood, or from an encounter that happened by accident rather than by design. The deep ocean covers most of this planet. Its volume below 1,000 feet is a space where most Earth species cannot survive, where human presence requires millions of dollars of engineering support, and where the animals that do live have had hundreds of millions of years to develop solutions to survival that land biology never needed. The Deepstaria found a way to hunt without teeth or tentacles. The immortal jellyfish found a way to avoid cellular death. The mimic octopus found a way to become 15 different animals. The Artemis limpet found a way to grow a skeleton that fits wherever it lands. Each discovery came from looking somewhere science had not looked carefully before, or from looking at something known and finally seeing it clearly. The remainder of the ocean floor, the zones not yet visited, the depth ranges explored only by sonar rather than cameras, the open water columns between the seafloor and the zones where research vehicles concentrate, represent the largest unmapped biological territory on Earth. They have supported life for far longer than any terrestrial environment. The organisms in those zones have had more time and more pressure to develop the kind of solutions that make biologists stare at footage and reach for the phone to call a colleague. Every new generation of deep-sea technology, each improved camera, each more capable autonomous vehicle, each better designed containment system, extends the range of what humans can observe directly. Each extension has produced surprises. There is no reason to expect that pattern to break. The Deepstaria floats somewhere in the dark right now. Its bell is expanding and contracting in water a human could not survive. The isopod is anchored at the center of the canal network, waiting for the next meal. The geometric web of channels spreads across the translucent tissue. The whole thing is nearly invisible in the total darkness of the deep ocean, 3 feet wide and completely alone. And whatever is sharing that zone with it, whatever animal with a body plan we have no category for yet, is floating right alongside it. We have not found it yet, but we are getting closer.